If you're looking to learn a bit more about the fundamentals of genetics, then welcome to Genetimix. My name is Tim, and on this channel we cover science communication, critical thinking, and, surprise, surprise, a bit of genetics. In previous episodes, we've covered the basics of DNA and RNA, and what genes are. If you haven't yet seen those episodes and need some more basic information on these topics, then I recommend watching those first, and then coming back to this one. All the episodes are in the same playlist, and the playlist is linked in the description below. Genetics is defined as the study of genes, genetic variation, and heredity, or inheritance, in organisms. It's one of the fields of biology that I describe as cross-functional because all living creatures have genes, and therefore genetics overlaps with most of the other fields in biology. There are many subfields to genetics, which include developmental genetics, or how organisms grow and develop, evolutionary genetics, species change over time, molecular genetics, which is the chemical nature of genes and DNA, and how genes function, population genetics, gene variation in groups, classical genetics, which are the visible results of reproductive acts, and epigenetics, which are heritable changes in gene expression that don't involve the DNA sequence alteration. As I mentioned in my earlier video on biology, there are many subfields to genetics as well, including things like microbial genetics, plant and agricultural genetics, human genetics, immunomedical genetics, forensic genetics, paleogenetics, behavioral genetics, genomics, quantitative genetics, statistical genetics, genetic engineering, genetic epidemiology, bioinformatics, and more. We'll be digging into most of these in more detail at some point. The purpose of this video is to provide an overview and to take a closer look at genetic variation, homology, heredity, and common ancestry. So let's take each of these one at a time and dig a little bit deeper. In the previous episode on genes, we already touched on alleles, which are variants of the same gene within a population, and looked at the blood group gene ABO as a simple example. We also had a first look at alternative splicing, where a single gene can produce multiple protein products, or isotopes, and how even a single protein can have multiple functions. These are starting points for a wider look at variation. We can add to this that many traits displayed by an organism are a product of complex relationships between many genes and their products working in tandem. Many genes will differ in their volume of gene expression from one tissue to the next. Many genes have a purely regulatory function. Their purpose is to regulate the expression of other genes. There are also many ways of altering the mRNA transcripts and controlling the amount of translation that is happening. There are also epigenetic factors at play, so-called because they are beyond the genes, such as methylated gene sequences, another form of gene regulation. And all this will differ somewhat between individuals in a population. There will be subtle differences in hundreds of these pathways. Unless a population has undergone a near extinction event, there will usually be quite a lot of genetic variation already present. Within this background, novel variation or mutations will also occur. There are quite a few different types of mutation. The simplest of these is the single nucleotide polymorphism, or SNP. This is where a single nucleotide is changed in the sequence. These are usually caused by DNA copying errors during cell division. If a SNP takes place outside a functional region of DNA, it will likely have no effect. This is called a neutral mutation. In some cases, SNPs will even be neutral in coding sequences due to the redundancy in the mappings of the codons to the amino acids, as there are 64 codons for only 20 amino acids. Even a change in amino acid might have a very limited effect on a protein, as many amino acids share similar chemical properties, and the function of the protein could be unchanged, or potentially very slightly worse, or even very slightly better. In addition, if a protein is an enzyme, only a small subset of the amino acids in that protein will contribute to the active site, the part of an enzyme that performs the catalytic function. Mutations elsewhere will have less effect than those on an active site. Mutations that cause negative effects are called deleterious mutations, and beneficial mutations are ones that improve the function of the protein. The next group of mutations are the indels, which are either insertions or deletions. As the name implies, these mutations include the addition or removal of one or more nucleotides to a sequence. In this simple example, we have a GA sequence inserted relative to this particular sequence or deleted relative to this one. If indels occur in a coding region, the effects are usually deleterious to the existing protein, as these can cause a frame shift in the coding. You will remember that each amino acid is coded for by a triplet of nucleotides, so a 2 base pair insertion like this will shift everything along by 2. This would result in a completely different amino acid sequence downstream of the mutation site, potentially even introducing a premature stop codon, shortening the protein sequence entirely. The impact of an indel depends on the gene. Some genes have 2 different alleles, and it's sufficient if only one is functional. Other genes require both alleles to be functional in order for sufficient protein to be produced. An indel in one of these in a germ cell could even render a fetus non-viable, for example. The next major category of mutations are duplications, where a larger section of DNA is duplicated. In this example, this whole section here is just copied out again. This often results in a whole gene being duplicated in its entirety, and this is one of the main mechanisms that creates new genetic information in the genome. The copied gene is free from selection pressure, and other smaller mutations, such as the SNPs and indels mentioned earlier, can now occur on this gene copy without any negative effects on the organism. Most of these gene copies degenerate into useless pseudogenes, but across a population some will accumulate new functions and become new genes. It's also possible for the entire genome to be duplicated. This will be a topic for a future video, but I will note here in passing that this is the single largest way of increasing the amount of genetic information available. Another common source of variation is the endogenous retrovirus, or ERV. Retroviruses get converted from RNA into DNA and are essentially randomly inserted into the genome. Cells that survive this kind of viral infection can find themselves with yet another source of new genetic information. Once established in the germline, these ERV sequences can change over time due to smaller mutations and become useful in their own right. A substantial proportion of the DNA in our genome, approximately 8%, consists of ERVs. Remember that the exome, the coding part of the genome that codes for all of our proteins, is only around 3% of the total. Here I'd like to go back and have a closer look at a gene. We started with this simplified model. Now it's time to expand on this model a bit and take a look at how genes are controlled. In the original simplified version, we had a look at exons and introns. When we take a look a bit wider at what's going on with the gene, we can see that there are both upstream and downstream regulatory sequences present for most genes. Upstream, or 5' to the gene, is the promoter region, which contains DNA motifs that tell the enzymes that produce RNA, which are called RNA polymerases, where to start reading from. Any random stretch of DNA can, in theory, be transcribed into one of 3 reading frames. There's 3 possible transcripts, which could then be translated into 3 different amino acid sequences. Transcription only happens when a promoter region tells it to start. However, promoter regions themselves are just DNA and can also be duplicated into other parts of the genome. This can cause totally new transcripts to be produced with their associated protein sequences, and this is yet another potential source of new genes. Our next topic is homology. Homology is a similarity between biological features in different organisms that is due to a shared ancestor. You may be familiar with common anatomical homologies like the bones in the forelimbs of all vertebrates. Homology is also something seen in genetics. We are able to compare DNA sequences using clever bioinformatics algorithms and assess their level of similarity. Homologous genes come in 2 main varieties: orthologs and paralogs. Paralogous genes are genes within an organism that have come from a gene duplication event. Orthologous genes are the same gene in different organisms that come from a common ancestral gene to both. In other words, the common ancestor of both organisms had this gene, and both have inherited it from the common ancestor. Many genes are both paralogs and orthologs. They were duplicated a long time ago in a common ancestor and then inherited. So how can we tell if a gene is homologous? As mentioned in the episode on genes, all genes have a structure with promoter regions, exons, and introns. If two or more genes have the same length with the same size of their introns and exons, and have similar sequences and similar protein products, then they will be homologs. The chance of two completely unrelated genes converging on the exact same structure and very similar sequence is extraordinarily low. So having introduced the concept of homology, we're going to move on to talk a bit more about heredity and common ancestry. All life on this planet uses DNA. All life is based on cells. The mechanisms for life are universal across all living beings. We introduced the concept of heredity in the prior episode on genes. All organisms inherit their DNA from their parents, and they in turn from their parents, and so on. Basic ancestry from one generation to the next is trivial. But what about common ancestry between different species? The species concept often causes confusion. What exactly is a species? How is it possible for a new species to form, given that every organism that is born has a parent of the same species? For larger multicellular organisms such as ourselves, a species is usually defined by its ability to reproduce with other members of the same species. This is called the biological species definition, and we'll stick with this definition for now and take a closer look at how a distinct species can form, a process called speciation. It's worth noting here that this definition of the species doesn't cover unicellular organisms such as bacteria, because they don't reproduce sexually. We'll come on to have a look at them in a future episode. Speciation was also, of course, the subject of Darwin's famous On the Origin of Species. He was the first to properly elucidate the concept that we all share a common ancestor. Darwin called this process descent with modification. Nowadays, we call it the theory of evolution. One possible mechanism for speciation is when two parts of a population become geographically separated. Over time, small differences in their DNA will accumulate, using the mechanisms described earlier, and eventually the differences will accumulate to the point where the two populations are no longer able to reproduce with one another. Speciation has occurred. There are now two species where before there was only one. These two new species both share a common ancestor species, the original species we started with. Speciation for longer-lived species can be difficult to observe directly because the process happens slowly over a number of generations. With that being said, scientists have now observed dozens of speciation events, both in the lab and in the wild. A particularly nice recent example is a rapid speciation event observed in Darwin's very own finches on the Galapagos Islands. I've linked the article for this one in the description below. Other prominent examples include various insects, including butterflies, fruit flies, and apple maggot flies, and we've observed many new species of plants. Since Darwin's time, we've discovered a huge amount of additional detail about how this process takes place, including the elucidation of the structure of DNA, the nature of genes, and the sequencing and phylogenetic analysis of millions of genomes from thousands of species. We can compare chromosomes and note that the number and length of the chromosomes and the position and orientation of the genes on each of the chromosomes is more similar between closely related organisms than more distantly related ones. We can compare the gene sequences, again noting that on average, the closer organisms are related to one another, the less differences we find in their orthologs. We can also use homology to make some interesting predictions. We know that many gene duplication events lead to degenerate pseudogenes, genes without function. If common ancestry is true, we predict that closely related species will also share multiple pseudogenes, as these will be inherited even though they are non-functional. We would also predict finding pseudogenes in one species with functional orthologs in others. A nice example of both of these at the same time are the GAPDH pseudogenes. While the main GAPDH gene is functional in both humans and rodents, rodents have hundreds of additional GAPDH pseudogenes compared to fewer pseudogenes in humans. Many of these individual rodent pseudogenes have human orthologs that are also pseudogenes. We can also make predictions about ERVs. Given the prevalence of ERVs in our genome, we predict that many of these are ancient infections that occurred in our distant ancestors. and there we will find very similar ERV sequences in the same places of the matching chromosomes of our most closely related animal cousins, and that these sequences will differ the more distantly related a species is from us. Chimps and humans, for example, share almost 100,000 ERVs in matching locations. The chance of even one of these being in the same place, by chance alone, i.e. from two separate viral infections, is extremely low. And again, there are many other examples in our genome, as well as the genomes of other animals. For anyone who doubts common ancestry, why would we share useless gene sequences and ancient viral sequences with other organisms in the exact same locations on the matching chromosomes? There are only two viable hypotheses. Either there is a designer out there who has carefully arranged all the genetic and other evidence to make it appear that we share common ancestry with other creatures, or we simply share common ancestry with other creatures. The second of these hypotheses is more parsimonious, so we will use Occam's razor and dispense with the former. We humans are apes and mammals and tetrapods and eukaryotes and living creatures, and we share a common ancestry with all other apes and all other mammals and all other tetrapods and all other eukaryotes and all other living creatures. And this common ancestry is written all over our genome and all over all of theirs. The amount of genetic evidence supporting common ancestry is truly overwhelming. I will come back and take a closer look at all the evidence for evolution, including a deeper dive into the genetic evidence, in a future episode. I'm also planning a step-by-step walkthrough of evolution. And that wraps us up for today. Don't forget to like, subscribe and comment. My social media links and Patreon are in the description below. Remember, science is amazing. Thanks for joining me, and I'll see you again soon.